The overall goal of the following experiment is to identify the exact positions of integrating viral vectors in the genome. This is achieved by an initial linear PCR reaction with biotinylated primers to enrich for vector genome junction sequences from genomic DNA on solid phase. In a series of reactions, double-stranded DNA with known DNA sequences on both ends of the product are generated, which allows for exponential amplification of vector genome junctions. Next, sequencing adapters are incorporated to LAM-PCR products in order to sequence and characterize the unknown flanking DNA. With deep sequencing, these fragments reveal the exact positions of vector integrations. The result is a diversity of amplified junctions as viewed by gel electrophoresis.
This method provides insight into viral vector integration site distribution in clinical gene therapy patients. It can also be applied to other studies such as insertional mutagenesis screens, virus infection, cancer and stem cell clonality or T-cell diversity. The procedure will be demonstrated by Kutschera, a technician from my laboratory.
For this procedure, first prepare the linker cassettes. Mix 40 microliters of each of the two LC oligos with 110 microliters of tris hydrochloride and 10 microliters of magnesium chloride in a microcentrifuge tube. Next, set the mixture to incubate on a 95 degrees Celsius heat block for 5 minutes.